Welcome to the Curiox Biosystems webinar. Our speaker is Dr. Sue Reynolds. Many of you in the Flow Cytometry community will immediately recognize Sue's name. She brings over 25 years of experience in the life sciences market with extensive background in flow cytometry and other cell-based assays. The presentation will last approximately 30 minutes. Please type your questions in the question box and they will be addressed at the end of the session. A recording of this webinar will be available at curiox.com. Allow me to introduce Dr. Sue Reynolds.
Thank you, Sheila. So I want to say thank you to everyone who's attending this morning. Hopefully this will be a fun and informative webinar. We've all worked with very challenging samples in flow cytometry, myself included. And there are some challenges with those. So hopefully today we'll discuss a few of those challenges and some new solutions to try to circumvent some of those challenges. So with that being said, there are several factors that can influence consistency and reproducibility and generating very optimal flow data. And as you can see here in this pie diagram, there's about 3 areas that can contribute to this. The first one is sample preparation, obviously. Then there's sample acquisition, and then we have data analysis. And it's in this sample prep pie, the blue pie, that is many experts in the flow field, including myself, say that manual sample prep is the key to reproducible quantitation, and that there can be issues that can arise with manual sample prep. And in a long-time friend of mine that I've had and have admired, Dr. Howard Shapiro, who many of you know in the flow community. Howard originally termed garbage in, garbage out, and I love that term. And that exactly means that if your sample is compromised in any way, shape, or form in the very beginning through sample prep, no matter how good you are at setting up your instrument and analyzing your data is going to be compromised. So it's best to get sample prep nailed down, get good recovery and good viability. And I know this can be a challenge in working with many sample types. So during the course of this presentation, I'm going to address some potential issues that arise during sample prep and how we can minimize their impacts on your data. So working with complex samples can lead to a lot of different areas of issues. We can get a generation of increased amounts of cellular debris, which will increase the nonspecific binding issues and can also lead to an increase in autofluorescence. We can also have poor cellular recovery and low viability. This can lead to a lack of clean separation between your positive and negative cell populations. Plus, the addition of mechanical stress that is due to repeated centrifugation cycles can compound these issues as well. And as you guys may know, and I know I've experienced this myself in my 25 plus years of flow cytometry, all of these above can lead to potential data loss. And you may ask, well, Sue, how can this lead to potential data loss? Well, what can happen is you can get a decrease in your staining resolution. You can get an increase in your data spread. And all of this in turn will decrease the resolution of your dim populations and will ultimately affect the reproducibility of your data. So I want to move on to a couple of challenging sample types that I know I've worked with, and I know some other people that I've worked with during my flow career in the field have worked with, and where some of our collaborators have worked with as well. So the first one, and I think many of us can agree, we've all been challenged with tissue samples. Okay, how many times has somebody come to us and they want to run an experiment with solid tumors? And as we all know, flow cytometry was not really generated for solid tumor type. It was always generated for working with cells in suspension. So obviously, solid tumors have to be dispensed into single cell suspensions. But in order to do this, you have to use dissociation methods, whether they be enzymatic or mechanical. And there are extremely harsh protocols on these cells. So if you're working already with compromised tissues, it's going to affect the recovery of these. It's going to affect their viability, and it's going to affect their cell retention. And a good example of this in the field right now is TIL recovery. Whether it be in an animal model system or a human model system, TILs can be problematic to work with. There are not many TIL cells within a tumor itself. They're a relatively rare cell population. So you're starting with low numbers to begin with. And then as you put them through these dissociation protocols, you're going to lose even more cells and viability. Infectious disease sample. This is kind of ironic with all that's going on in the world right now with COVID, but any of us who have worked with infectious disease samples in the past in flow cytometry, especially in the area of sorting, we have the potential of generating aerosols and droplets that are infectious. So those can be problematic to work with as well. Blood. Well, you can say, Sue, how is blood a challenging sample? Well, it can be if you're working in a small volume. Think of those of us who are working in mouse samples. On the best of days, mice do not like to give up the goods very readily. So you might get at best 500 microliters of blood to do an experiment. Also, if we're working with neonatal or pediatric samples, they can be challenging as well. Again, small blood volumes. Or if we're working in clinical trials where we've had maybe only 1 or 2 tubes of blood drawn and it's being parsed out in a lot of different clinical assays, somebody may come to us and say, you know what, Sue, we've only got like half a mil of blood to work with and we want to look at all these panels. So it can be a challenge to work with this. We also can work with sensitive subset populations. And what I mean by this is just by handling them themselves can really be disruptive to either their recovery or their viability. Monocytes, neutrophils, granulocytes, these are all samples that come to mind that I've experienced working with or talked to others who have had issues working with. And finally, and I think this is really important aspect as well, is cells can be very fragile prior to processing. Clinical samples, if the samples are not frozen correctly at the clinical site, when they arrive at the lab and they're thawed out, you very possibly have extremely low viability and cellular retention. And also, I'll show you some data from one of our collaborators where certain animal models themselves are fragile, and that in itself makes the cells that you're working with fragile as well. So just to kind of give you an example of what I just talked about, I'm going to show this data in more detail later, but this is tumor processing out of TIL samples from a fragile mouse model system that can lead to issues just through some traditional platforms that we've used in the past. And one of those traditional platforms has been centrifugation. I mean, we've used it pretty much our entire career because that's what we were trained on and that's what we've been used to using. So as we know, proper cell washing is crucial to achieving very good cell purity of your sample. And this is especially true when you're using small sample volumes or you're running high sensitivity assays. Proper cell washing, as we all know, leads to increased cellular resolution because we're getting rid of all of our debris or unbound antibody. And this in turn will yield accurate target cell identification. So, as we all know, and I've said, centrifugation is a very well-established method, and we've used it for a long period of time. And usually with those samples that we've had the luxury of working with a lot of cells, myself included. I've been a T-cell immunologist my entire career. So working with these samples can be problematic. But recently, over the last several years, we've been working with a lot of assays that require a few cells or maybe a single cell for a downstream application. So what we need to do is we need to minimize these challenges that arise during the centrifugation process. And what we need to do is try to increase our cell viability and our retention. And we can do that by trying to use more gentle and natural sample prep methods that will ensure that your data is biologically relevant. Okay, so all of you are probably scratching your head right now and saying, Sue, how is this possible? And I'll get to that in a minute. So what I want to show you is what centrifugation can actually really do to your cells. It can cause a lot of cell damage. it can cause cell loss, and it can cause a bias in your data. And what I want to show you here is, this is a nice picture of cells that were taken by immunofluorescence. On the left-hand panel, we have cells that are processed by centrifugation. On the right-hand panel, I can show you cells that were processed not using a centrifugation method, but using a laminar wash technology. And what I want to point out here with these two panels is, on the left, you can see we have a fair amount of debris. We have a lot of cell loss, and we really have low expression of whatever endogenous or exogenous proteins that we are looking to stain, compared to the panel on the right that has not undergone any type of centrifugation method. Now, just imagine for a minute if this was a very precious CAR T sample, and you can't take these kind of losses with your cells when you're looking at this. So these cells can be lost or destroyed, as I said, due to either the mechanical stress of the centrifugation alone in combination with a tedious workflow. So what I want to show you right here is a comparison of looking at this non-centrifugation method, or the laminar wash method, versus centrifugation. And this is working with some very compromising cells that we tend to work with in flow cytometry, looking at monocytes, neutrophils, and NK cells. As we can see on the top row, we get nice retention of these very finicky cell populations. And on the bottom, you can see how the centrifugation has really taken its toll on these, where we're not getting clear separation of our populations, and of a lot of our rare populations, or populations that are very prone to handling issues, are disappearing. So I mentioned the laminar wash technology for a centrifuge-less sample processing method. And basically, let me introduce you to this really briefly right here. As you can see on the right-hand side, it's a two-process system. We have a plate, and we have the washing system as well. The plate, the laminar wash plate, is an array of hydrophobic spots that are surrounded by a hydrophobic surface. And what this creates, it creates a virtual wall that separates the spots. So instead of having a traditional walled plate like we're used to working with, these show virtual walls. And I'll give you a view of this in a moment. So the plate washing is faster and provides more complete washing. So a 9-cycle wash for 2 minutes is equivalent to 3 washes in a centrifuge. There's no flicking of the plates, which in itself is an art. So I don't know if any of you have tried this, but I've done it myself. I've seen other people do it, where people flick, they get nervous, they back off, they flick again. So now they've got residual fluid, they're losing their cells, or they flick and then they tap their plate onto like a diaper that's on the lab to get rid of any extra fluid. And when that happens, that can drain cells out of your well and onto that diaper. So by not having this flicking procedure, we get improved data reproducibility and consistency in the washing of our plates that contain our precious samples. The system also has increased operator flexibility with comparison to a centrifuge. Depending on the application, the user can adjust the wash cycle and the flow rates for the particular application and the cell rate or cell type that they're working with. So all of this leads to improved workflow with less hands-on time. And what does this really translate to? The multiple steps that we've always done with a centrifuge is now reduced down to 1 step using the laminar wash system. And I'm not sure about those of you on the phone, but in my entire career, if I got paid for every single time I was looking for a centrifuge to spin down, I'd probably be retired and living in some exotic island right now. So I wish this technology had been around when I was doing flow cytometry, because to be honest with you, I'm a lazy washer. And if I can get away with cutting washes or not washing, I would love it. So I would have loved this technology to be around during my early part of my career. So with that being said, I want to focus on the plates with you guys for a minute. So here's the plate up close and personal. As you can see in this larger view of the plate wells here, these plates are uniquely engineered, so we can maintain each sample as a separate droplet, as you can see here. which is held into place by surface tension and hydrophobic interactions. So you see these two little nodules, hopefully on either side of this blue well that I'm showing to you. So two nozzles are lowered onto each side of the well, one on each side, one for dispense and one for aspirate. The nozzles themselves initiate the laminar flow by dispensing and aspirating the buffer on the opposite side of the well. The lateral flow that this process initiates will decrease the turbulence that's placed on these cells, and will lead to increased cell retention. So the wash cycles basically remove all the debris, unbound antibodies, whatever it is, leaving the cells on the bottom intact and unperturbed. So this is the key feature of the Laminar Wash system, that the cells settle to the bottom of the well, they go through these washing steps, and they remain in an unperturbed state. So what I want to do, oh, sorry about this. I want to just show this to you really quickly on the right-hand side, and then I'll show you a video of the whole system working. But on the right-hand side, I want to show you before washing. In the first row here, that's really blue, that's food dye only. And what that represents is it represents any debris or antibody, unbound antibody that could be in your samples. The second row over, that's kind of an aqua color, that's a combination of food dyed and beads. So the beads are yellow, that would be equivalent to cells. The next row over that's yellow is beads only, which would be cells themselves. And then the last row is just a row that contains, the clear row that contains buffer. So after washing, you can see that the blue dye, food dye, has been removed, which means that the debris and the antibodies have been removed, leaving behind the yellow, which are the beads, which is that would be your model system for your cells. So again, just showing you that we've efficiently washed all unbound antibodies, debris, particles, whatever away, but have left the cells intact. Now I'm just going to, hopefully this will work, where I can show you a quick video. Here we go. This is loading the plate into the washer. As you can see, the dark blue representing debris or antibody. These are the nozzles. As you can see, they are aspirating and dispensing the liquid onto the plate. So it goes through how many wash cycles that you've programmed the instrument into. And then when it's done, the plate is then comes back, and here we go. Here's the plate. After any of the unbound antibodies, debris, unbound proteins, whatever we're working with, are washed away, and we're left with just the yellow wells, which would be those wells that contain our cells. Let's take a closer look at this process and what's going on. And here's a little cartoon that I like, as you can see. And this is just what happens during the process of incubation. So here we've added our cells to the plate with antibody. And during this time where the cells are incubating with antibody, the cells will settle to the bottom of the plate surface via gravity. So you can see this here, where we add the cells over, say, a 30-minute period of time for this particular sample. The cells settle to the bottom of the plate. Then through diffusion, the unbound antibodies or unbound proteins or whatever is in there are washed away by the laminar flow, leaving the cells on the bottom that have our antibodies bound to them. I mentioned this before, that the laminar wash velocity is greatest at the top of the well, washing away the unbound antibodies and the cellular debris. Again, leaving the cells intact, so that velocity is almost static at the base of the plate. So that is why the cells are unperturbed. So this lateral movement is what washes everything away. So now that I've kind of introduced you guys to the technology, what I want to do now is show you some examples of some data that's been generated by some of our collaborators in working with some of these challenging samples. So the first set of data that I'm going to show you is from a collaborator, Dr. Eberly and Charles River Labs here in Boston, Massachusetts. He has kindly provided us with some data working in a tumor mouse model system for looking at immuno-oncology and oncology therapeutics. So this tumor processing model that he has showed with us is one of those models that I had alluded to in the beginning of the talk that can be very fragile in itself. These are B1-6F10 inoculated mice. They are a common model that's used for oncology research, but the animals themselves and the cells in the animals, excuse me, while they're being treated are very, very fragile. So on this upper panel here, what you can see is this is manual centrifugation. As you can see on the far left, on the forward scatter, side scatter dot plot, you can see all the debris that's there, and it's very difficult to draw an accurate forward scatter, side scatter gate on your lymphocytes. Singlets aren't looking all that much better, and we really don't have a lot of live cells either. And what this happens to your data is you've compromised your lymphocyte staining. So for CD45 positive cells, we're only looking at about 10 or 11%, and that only translates to about 17% of CD3 positive cells. This is in contrast to the Laminar Wash system here at the bottom, where you can see there is much less debris in the sample using this type of washing technology. We get a very nice separation of our lymphocyte population where we can draw a nice accurate gate around these. And we've gone from about 7 to 8% in this gating population into about 26%. We also have a cleaner singlet population, and basically look what this translated into. I mean, we've got almost a tenfold or fourfold increase, or 10 to fourfold increase in our live cells. We've gone from 9% up to 48%. And this is translated in, if you look over at this next plot under CD45, we've gone from 10% CD45 positive cells up to 91%. And look at that nice separation and the retention of your cells. And again, this translates into your CD3 positive staining. So you can see that the Laminar Wash is really showing enhanced improvement in TIL immunophenotyping. So this is showing in a mouse model system, but you can also imagine that this would hold true for human model systems as well. So think if you're getting tumors from a clinical trial, from patients, again, the retention is going to be better if you use a gentle, more gentle washing method. So while we're speaking of the mouse model system, I just want to show you again some data from Charles River, again from Dr. Eberle. We're working with small volumes of blood, and here he combined Laminar Wash with magnetic bead depletion in murine blood samples. So you can see here there are two mouse model systems that are commonly used, VALB-C and C57. He looked at 25 microliters of blood, and basically what I want to point out here is by combining these two methods, small blood volumes can be handled consistently without the harsh RBC lysis and centrifugation methods that are going to cause decreases in your cell retention and your cell viability. So now I'm going to move on into the human world a little bit and look at the performance of the Laminar Wash when compared to centrifugation in rare cell populations. So in human blood, another challenge that arises is trying to deplete red blood cells from whole blood samples. So traditionally, we have lysed RBCs, washed them, and centrifuged them to remove them. So I don't know if any of you really thought about this or whether you already did know, but the lysine solution itself, even though it's not in there for a very long period of time, can be harsh to the cells. And it becomes very problematic again if you're trying to retain and resolve rare cell populations. So here in this particular study, we took 100 microliters of blood. We stained it up with a 16-color immunophenotyping panel that was focused more on T cells. And we compared the Laminar Wash to centrifugation and magnetic bead depletion to ammonium chloride lyse. And what I'm showing here on this top row, this is RBC depletion using the magnetic beads with Laminar Wash. The middle panel is using traditional ammonium chloride lyse and then using Laminar Wash. And then the bottom panel is our traditional ammonium chloride lyse followed by our traditional centrifugation methods. And as you can see here, when we look at the populations of cells, a rare cell population like these mate cells, gamma delta T cells, and Treg cells, we get a nice clean separation and high retention and resolution of these populations using the combination of bead depletion and laminar wash. We still get a pretty good retention and resolution when we use ammonium chloride lysis in combination with laminar wash, although it's not quite as good as you can see in the Treg cells. And finally, if we do the final comparison with ammonium chloride, look how we're losing these really precious populations of gamma delta T cells here and in the regulatory T cells. This also can become an issue if we want to look at activation markers. And that's shown here where we took that same population of cells and we're drilling down through the memory T cell response and looking at the CD4 and CD8 memory compartments and looking at activation markers. And in this particular experiment, we're focusing in on CD27 as the activation marker. And again, you can see here the nice resolution using the bead depletion and the laminar wash for our double positive population of the memory cells and for the activated cells as well. There will be differences between the CD4 and CD8 populations. Again, here this is showing the ammonium chloride lyse. CD8 seems to be very affected by the ammonium chloride lyse. CD4 a little bit, but down here we're getting a lot of depletion and of our memory CD8 positive T cells. And this is not unusual. Different cell types will become more fragile than others in memory cells, but this becomes very important in our experiments, and especially if we're looking at a polyfunctional immune response, either in cell therapy, immuno-oncology, or these days they're becoming a target for COVID samples as well. So that kind of leads me into my next slide with COVID. I just want to show you the performance with some infectious disease samples that I'm working with. So back this past March, Curiox got into a collaborative study with stem cell technologies where we were looking at infectious disease samples and vaccine development and how we can handle these samples in a safe and efficacious manner by combining these two systems together. So that's exactly what we did here. These instruments can be placed in a biosafety containment hood. So that way the blood sample processing is contained all the way through from start to finish with your sample. So you have no washing with centrifugation. So you can imagine that we're eliminating the potential for aerosolization of infectious particles. And again, this washing method yields very superior data analysis, as I've shown you on the previous slide, and I will show you again in another example of this data working in infectious disease. The cells aren't stressed by lysis or centrifugation, so therefore we've got improved staining resolution. So a lot of times when we do these experiments, we don't think of the additive effects of handling these samples. Okay, I'm lysing them, I'm centrifuging them, I'm perming them. You know, all of these are very harsh methods on cells, and as we work with them and manipulate them, we're going to lose. The number of cells are going to decrease as we work with them, and their viabilities are going to decrease as well. So as you can see in the schematic right here, this was a relatively quick protocol. About 20 minutes for the depletion of the RBCs, a 30-minute stain incubation, a 5 to 6 minute wash, and then 8 to 10 minutes for looking on the flow cytometer. So as I mentioned, in COVID vaccine development, neutrophils and monocytes are becoming a huge population of interest. And both of these can be a challenge just in themselves through sample handling to work with. And since there a great interest in COVID-19 studies, I just want to share some data with you that shows better resolution and retention of these populations using laminar wash. So as I had stated in the previous slide, this particular experiment was done in a very similar fashion where we took 100 microliters of blood and we compared the magnetic depletion to the ammonium chloride lyse, and we compared traditional centrifugation with the laminar wash. And then we stained and focused in on our neutrophil and our monocyte population. And I want to draw attention to this graph over on the left-hand side or the right-hand side of the slide, where we can look here above on our CD45 population. If we deplete with magnetic beads and do the laminar wash system, we have very high retention of our cells as compared to our lysing method, either using laminar wash or lysing and using centrifugation. So again, just showing we're retaining the bulk of our lymphocyte population through this process. Now, if you look at the graph down here on the lower right, on the left hand, or on the neutrophil side, on the left-hand side of these graphs, you can see if we deplete with magnetic beads and then do a laminar wash, or if we lyse and do a laminar wash, our percentage of our neutrophils is really good. It's about 80%, but look how much we've lost if we just do an ammonium chloride lyse and a centrifugation step. We're down to about 10%. Same with the monocytes over here on the right-hand side. If we deplete, do a laminar wash, we retain the bulk of our monocyte population. And again, if we lyse and then do a laminar wash, or if we lyse and we centrifuge, we're losing a lot of our cells. So as this data shows, we can safely and more reproducibly obtain results with increased retention of both our monocytes and our neutrophils using the magnetic depletion method from stem cell technologies in combination with the laminar wash. This is a more gentle protocol as compared to the more traditional methods that we've used with centrifugation. So hopefully this morning I've showed you a couple of things. Hopefully I've showed you a new way to think about how you treat and process your cells, because I don't think we think about new ways. You know, we stick with the old ways because it's tradition. But the laminar wash really streamlines your sample prep process. It's time saving, as I've shown you. You know, we can do in one wash replaces 3 centrifuge washes. The instrument and plates are very user-friendly. They're very easy to operate. Because we're semi-automating this process, we're taking a lot of the operator and location to location variability out of your results. So now your results become a little bit more consistent because we're not depending on operators or locations. I've shown you, at least on the safety side, that we get complete containment in a biosafety cabinet. So those will all help improve your workflows. And by improving your workflows, you're obviously going to get higher quality data. So back to the first slide that I showed you of garbage in, garbage out. So we have increased fidelity of rare and native cell population frequencies. I think I've shown you, especially in that last slide with the monocytes and the neutrophils, that we have higher cell viability and retention. In the slide that I showed you from Charles River Lab with the mouse till samples, I showed you that with enhanced debris removal, we've enhanced our capabilities of really honing in on our rare till sample population. All of this ultimately leading to cleaner data with less staining background, which gives us higher signal to noise ratio, and ultimately gives us that clearer resolution of distinct populations, which is really, really important because many of us are working in areas where these populations are very, very dim, or they happen to be a smeared population. So really trying to decipher them and hone in on them can be problematic if our samples are not clean and if we don't have a lot of cells to actually run through the flow cytometer and get accurate data accounts on. So I'm just going to finish up here by highlighting some of the benefits of the laminar wash technology. And we've talked today about flow cytometry and immunophenotyping. I've shown you how we've had higher cell retention, better viability, less debris. I've showed you with infectious disease and vaccine result that we've got improved biosafety because we've not only eliminated the centrifugation step, but we can put the washing and the instrument itself, those steps, in a biosafety containment hood, thus decreasing the aerosolization potential. I did not go over this in this particular talk oops, I'm sorry. But this process has been used in single-cell genomics and proteomics and proteogenomics, where again, they need higher cell retention and doublets, especially if you're doing single-cell sequencing, can lead to background issues if the cells are not washed thoroughly enough. So using the Laminar Wash technology gives us a higher retention of our singlet population and gets rid of our doublets. And I have talked about automation as well. You've seen how it can be partially, or we do have instruments that are completely automated as well. So I just kind of want to introduce you to the family of the Curiox instruments. There are 3. On the upper left, we have the Mini. This is a small little instrument that does 8 wells at once in a 16-well strip. It's got a very small footprint. This instrument can fit into a biosafety containment hood as well. We have our workhorse, which is the HT2000, which is capable of washing 96 plates at once. Again, being able to automate the washing skills, and that's what I've shown you today with all the data. And on the right-hand side is our fully automated system. So this is an entirely automated cell prep platform from start to finish. So it not only washes your cells, but will stain them as well. And it's ready to go right out of the box. And we have several major pharmaceutical companies who are currently using the Auto 1000 for many of their therapeutic target areas. So I want to thank all of you, and I'm open now to take any questions that anybody has. Sheila?
We do have some questions that have come in. The first one, would you be able to recommend centrifugation of live human cell samples, hematopoietic origin or human iPSCs, both live and fixed, tube versus plate?
Could you repeat the beginning of that question again?
Would you be able to recommend centrifugation of live human cells?
Right. Okay. For the person, we've traditionally done centrifugation for those types of cells, but I can tell you right now, we do have some collaborations going on when we are addressing using the Laminar Wash system with these cells. Again, we're in a situation where iPS cells can be fragile, and there's not many of them around. So you could do plates, you could do tubes. I've worked with iPS cells in my past life. Depending on where they are generated from, they can be difficult. I don't have any data to show you right now, but I can tell you internally we are working on using this system and adapting it to that particular cell type.
There is a little asterisk to this: centrifugation speeds and times. Any recommendations, I think, is the question.
At this particular point, I don't really have any recommendations for speeds and times on a centrifugation. Again, it's going to depend on the types of iPS cells that you're working with and what sort of shape they're in. I mean, if you're concerned, I would probably say, you know, less is better. Timing wise, and in the RPMs or G-forces that you apply to those cells, again, they can range. You would have to test those out for the iPS cells that you're interested in working with.
Thank you. Can laminar wash apply to E. coli cells?
Interesting question. We have not tried that yet, so I'm not really sure. I'm just hazarding a professional guess here that I would think you could probably use it for E. coli or bacterial cells, but it's something that we have not looked at either internally or our external collaborators have looked at either.
We have 2 of the same question. Impressive presentation. I'm now terrified to centrifuge. Could you please provide a ballpark idea of how much the Laminar Wash HT2000 costs?
Sure. So the HT2000 approximately, again, depending on options and what you get, it's approximately $60,000, $60K plus or minus. So I hope that helps. I'm glad I terrified you, because I'm terrified now too for centrifugation. I never really realized what I was doing to myself.
Okay, next question. What are some common difficulties in running the laminar wash, and what tips do you have to overcome them?
We haven't really come into a lot of issues running the laminar wash. I would say if there was anything that could be a common, I mean, not common, but could happen. Again, if you're working in solid tissue samples and you don't put them through a cell strainer prior to putting them onto the laminar wash system, you might get some clogging of the nozzles, but that can be easily averted by putting a priming system in there. But so far we have not had any issues with the system at all.
Great. What are the speeds used with the data presented?
That's a good question. So the cycles, I think for most of the data that was presented, the wash cycles were about 2. Each of those cycles lasts from anywhere between 2 and 5 minutes. I think the bulk of the cycles were for a 2-minute, and the flow rates, I believe, were somewhere between 5 and 10 microliters per second. So cycles can be repeated for anywhere between 2 and 20 times. They generally last anywhere from 2 to 5 minutes, and you can adjust the flow rates from anywhere from 5 to 20 microliters per second, if that helps.
Thank you. How many 96 well plates can you automate with the Laminar Wash Auto 1000?
That's a good question. I believe that the plates are done, I believe they're done one at a time, and I think you can put a plate loader onto that. I am not as familiar with the auto system right now. I'm just coming up to speed on the 2000, so I'm kind of speaking out of turn here. I don't know if any of my colleagues from Curiox, I don't know if they could answer that question in more detail than I can. Okay.
Okay.
Any other questions?
Let's see here. Yes. Can you run less than a full plate?
Yes, you can. So what you can do is, if say you want to run half a plate, just run half that plate. Then make sure there's buffer on the unused wells. Basically put it into a Tupperware container and put it in the refrigerator, and you can reuse the other side of the plate.
Great. Next question, can the laminar wash plates process incubations at different temperatures?
Yeah, on the auto system they can.
Great. What is the minimum time required for cells to settle during incubation time?
That's a really good question. So a lot of it's going to depend on the cell type that you're working with and what the density of your cells are. Generally speaking, for PBMCs, which we've done a lot of our work on, it's about 30 minutes. It might be a little bit longer for some other cell types, or if your cell number is a little bit lower. So it's about 30 minutes. Again, it's going to depend on the sample type and the cell number. And you can range anywhere in the system from 10 million cells. Again, in PBMCs, it can handle from 10 million cells down to about 1 cell per well. So what you'll have to do is you'll just have to fine-tune the settling time based on the sample number and the type of sample, but 30 minutes is kind of a general ballpark.
Thank you. And our last question is about pricing. We've answered, what are the rough price of the 3 instruments?
Right, good question. Mini is approximately $20K. We went over the HT, which is approximately $60K, and the auto runs at approximately $200K.
Thank you. One more that just came in. For intracellular staining, fixation and permeabilization steps can be performed directly in the plate as done for incubation with antibodies?
Correct. Correct. So when you're permeabilizing cells and you're going intracellularly, again, your settling times might be a little bit longer than 30 minutes, because remember, you're punching holes in these cells and they're basically becoming wiffle balls. So they kind of like to bounce around in solution a little bit longer. this even goes for centrifugation steps. So you might have to give them a little bit longer time to settle down to the plate just because they're a little bit more buoyant because they've had holes poked into them.
Do the nozzles for dispensing and aspiration of wash need to be cleaned, and are they disposable?
Good question. No, they're not disposable, but there are, when you set up the system and you prime it and you set it up and you close it down, there's a protocol that you need to go through to prime the cells and shut the instrument down that basically cleans all of those systems. So it's very much almost like the fluidics on a flow cytometer. So, you clean that out, you let it sit overnight. I believe you clean with ethanol, then let it sit with water. Then the next morning you have to reprime the system before you run.
Great. So that was our last question.
Thank you very much, everybody. Stay safe, stay healthy, and try to have happy holidays.
Goodbye, all.
Thank you. Bye.